Good evening, good afternoon, good morning. What order is that? Who muddled that order up? Anyway, it's evening here, so I started with that. And welcome to you, viewers, wherever you are and whenever you are, because most of you watch this not live, but afterwards when it's a podcast. Anyway, here it is, Free Thought Hour. Please say welcome to co-host Tercia. How the devil are you, Tercia?
I'm good. Thank you, John. Nice to be here. How are you doing?
I'm okay, thank you. I hope you've completely recovered from your ailments that you've been suffering from.
I'd say 90%. Yeah, I'm getting there.
Good.
Getting there. Yeah.
Excellent news. And say hello to our guest, Seth Garren. How the devil are you, Seth?
Hello, I'm great. Happy to be back.
Good.
Yes.
Good.
Hello, Seth. I want to Garren. Garren. What's the origin of your surname? Do you know where your ancestry?
It's like French, I think. My ancestors came from the Norman and England and bits of Northern Ireland, like around the era of the Mayflower. So I literally have descendants on the Mayflower, or ancestors on the Mayflower. So.
Ooh. Do you know there's a replica of that at a little port down in the West Country here?
No, I didn't.
A copy of the Mayflower. Yeah.
So that makes you American, not royalty exactly, but you're authentic American if you can say that you had ancestors on the Mayflower. Am I right?
Oh, the Amerindians would have something to say about that.
I would call myself one of the first generation immigrants.
Yes.
It's a bone of contention.
Yes. Seth is here because I bumped into him on social media, Facebook. We've been chatting before we started streaming, and we think it was the Unbelievable Show. Well, it's not a show on Facebook, is it? It's a site. But I noticed that Seth was sort of arguing, like I would argue, against some theistic statements and claims and assertions, which seems to be all they have. And so we struck up this to and fro, and it turns out that he's a really interesting scientist, genomics research, and it's sort of at the cutting edge of development of drugs, which is very interesting field to be in. I want Seth to tell us about that.
John, I absolutely want to, but I need to clarify something. What's the difference? I need Seth to clarify for me, ignoramus that I am. What is the difference between genomics and genetics, if there is at all?
So genetics is sort of a larger, broader catch-all term that has to do with anything concerning genes. Most of the time when you think of genetics, you're usually thinking in terms of specific genes, like you inherit a genetic disease. And once you've identified the gene, you're kind of looking into what to do about it. Things like genetic counseling, like, oh, we have a family history of this known mutation that causes a risk for a particular disease. Whereas genomics is the science of exploring the entire genome at once. So sequencing the entire human genome of somebody or an entire population of people, or sequencing every single gene simultaneously that is expressed by a genome in any given biological context. So
it's the genetics of an individual.
Like an individual gene. You can have genomics of an individual. Like, for example, if a close friend of mine is exploring a potential genetic disease because they have their child has been suffering from some mysterious symptoms, and they are doing what's called whole exome sequencing, where they take the 2% of your entire genome that directly instructs what a protein should look like, and they only sequence that part. And they're doing that to see if there's any hard and fast breaks in the instruction that would cause a malfunctioning protein that could potentially explain the mysterious symptoms.
So this sort of borders on the computer simulation of how proteins assemble into a shape, doesn't it? I mean, there's been an update to the algorithm for AlphaFold, hasn't there, recently?
Yeah, apparently they're adding whole new elements of artificial intelligence and machine learning to help make predictions about some of the structural features and how proteins fold. Typically, when you look at it from a genetic standpoint, we treat proteins like they're strings. Like it's just
Yeah.
a series of instructions. They have these amino acids in this order, and that's all that matters.
Mm.
And if you're trying to predict what'll happen in a biological system, that's usually enough. But if you're trying to understand, if I have this string of proteins and I want to know what shape they would make,
Yeah.
how can we predict that? And so, with a lot of the new machine learning, it's more than just the order of the amino acids. There's other things like what amino acids, if they were in a particular shape, would end up next to each other in 3D space. And
Yeah.
what they do is they use things like ChatGPT's uh, large language models to predict adjacent residues that would be next to each other if they form a known structure. And that helps
Mm.
kind of condense the seemingly endless number of combinations down into something that's more actionable.
Yeah. Yeah. Because it's not the necklace that matters. It's not the linear constitution of a polypeptide, is it? It's the actual shape that gives the function of that particular protein. Yeah.
So Seth, what does your day job entail? Do you work for a pharmaceutical company? Are you allowed to talk about who you work for and what you do? What does your typical workday look like?
Yeah, so I work for a large pharmaceutical company. I've actually it's my second, so I've only worked in the sort of big pharma space, but my job is basically to make sure that the scientists in R&D are making the best use of the cutting edge tools that are coming out as they come out, so that they can answer questions about what all is going on in a biological system when they're trying to either study a disease or study the effects a candidate drug has on a disease. So my particular specialty is RNA sequencing. So as most people might remember from high school biology, there's a thing called the central dogma, where you start with your genome, and then the genome is sort of like a library. And then you have the RNA, which is sort of like you go into the library and you pull out a book, and the book is, say, a cookbook, and it has instructions for how to make a recipe. And then you follow the instructions and make the recipe. That's like the protein, and then the protein does jobs around the cell. And so most of the stories we tell about biology is, what do proteins do in cells, and what can cause them to do that incorrectly when you have a disease. So for us, we basically start pulling all the books out of the library at once and read them all and say, okay, which books are open, which are not, and are there any books that look like they're making too much of a particular recipe within the cell that we could point to and say, aha, that could be causing the disease. And then we could then say, maybe we should take that one book and make it not produce so much as it used to, and then that can become a strategy for a new drug. Likewise, if we have a drug candidate and we wanted to know, well, what all happens when we put it in the cell, we can have cell culture models where you'll have like a bunch of cells in a petri dish. You can have animal models, or you might have mice, or even certain species of primates that we could add our drug candidate to. And then we can ask, well, what does this do in this tissue? So you might say, what does it do in the kidney or the liver or the brain? And then you can take that tissue and ask, what's the difference between the treated tissue and the untreated tissue, to see what other gene changes and biology changes are happening once you treat them with that, and do they have any safety risks, and do they give us evidence that it's doing what we hope it's doing, that it's in fact changing in the direction we want it to go.
Sorry, John, you go ahead, because I'm going to sound like a really unintelligent high schooler. Because
not at all. No. This is the point. We're trying to get people to understand. So, ask your questions. Indeed.
So it just so happens that John and I had a conversation earlier this week about. I don't know if you're familiar with John Campbell. I watched it. He's a retired nurse, and the long and the short of the story is that he claims, and he's not the only one, I must say, that the mRNA vaccines seem to have the ability to contaminate, and I'm using the word contaminate not in any sense to say that we're going to turn into aliens or monsters or anything, but to contaminate the DNA. And that's the question that I want to ask. I know that RNA stands for ribonucleic acid. DNA is something
Deoxy.
nucleic acid. Deoxy, that I can never remember. How does DNA and RNA relate? To your explanation about the library and the cookbooks or the books work brilliantly for me. So could you do something similar to just explain to me what the relationship is between RNA and then DNA, and then perhaps, if it's all right with John, you could enlighten us if you have any expertise or in that field, which you probably do if you work with in medicine or with pharmaceuticals. What is your opinion about the mRNA COVID vaccines and other mRNA vaccines? And are there any others in use?
Yeah, so the sort of the term's called the central dogma. That's sort of the thing that, like, the whole mitochondria is a powerhouse of the cell sentence that we hope everybody comes out of high school memorizing. In this case, with respect to the central dogma, it's DNA is copied into RNA, which is translated into protein. And the mechanism by which that happens is DNA is basically just a long chain of bases. You know, you have your ATCG bases in a what's called a heteropolymer, where it's not just the same sequence over and over again. It's very different sequences, creating all these different combinations. And what's nice about it is that it's also anti-parallel. So one strand goes to the right, the other strand goes in the opposite direction, and that allows it to preserve the information and copy itself. So if it gets split in half, then you can create two copies, and that's how DNA replicates. But what's also nice is that you don't have to copy the entire length of the genome. You can copy just like a little shorter segment of it. And what RNA is a short-term copy of a small segment of DNA that the act of copying it could be tightly regulated. And so the cell can control where in the genome that segment of its DNA gets copied into a shorter, typically single-stranded, what's called a transcript, because it's transcribing. It's like you're reading a book on one side and then writing every letter you see. With the exception that instead of having a T, RNA has a U. So if you have ATCG, it's AUCG for RNA. The other difference is that in the name ribonucleic acid, DNA is called deoxyribo, which means it's missing some oxygen, which ironically, that missing oxygen actually makes it more stable. But that's just sort of why we call it that, just in case people are curious. Anyway, so what the RNA does is it contains a temporary transcript that could then be exported out of the nucleus, and that transcript can be processed so that it can then provide as a template for enzymes that assemble proteins. So an enzyme they're called ribosomes, and the ribosomes will sit on the RNA, and based on whatever three letters it sees, it will then attach a specific amino acid in that order. And then as the amino acids come out, they have special proteins called chaperones that fold them. And then that makes your final protein, and then the protein can go do its job. So that's just the central dogma. DNA gets transcribed into RNA, which is translated into protein. That's the boilerplate message we want people to take from high school. Now, with the mRNA vaccine, what we're doing is we're basically taking a lipid nanoparticle, which is just like a little ball of fat, for lack of a better word. Like you get the little soap bubbles. It's like a little ball of fat that's like a bilayer of its own, and we can fill it with RNA of whatever sequence we want, and then we coat it with things like, in the case of the Pfizer vaccine, we literally coat it with laxative, something to kind of irritate the local area. So you inject that into like the muscle, and it will cause irritation in the local area, and then that will cause the immune system to show up. And then what will happen is the muscle will take up that lipid nanoparticle, and the lipid nanoparticle will have instructions for it to make a protein that is normally expressed only in the virus. So one of the things that cells do is the cells will constantly report to the immune system what proteins are inside of them. So they'll take some of the proteins and they chop them up, and they take pieces of those proteins and they present them on the surface of the cell. So when immune cells come by, they'll check and they'll see that protein. And if that protein is from something like a virus or from some potentially cancerous behavior, then that will cause it to kill the cell. Or it will take that information and it will go to a local, like a lymph node, which is basically like a police station. So think of it this way, like you're passing out wanted posters. So you say, hey, here's instructions for how to draw a police sketch of a suspect. And then a bunch of like detectives will stop by, knock on their door and say, hey, like, is there anything weird? They say, here, look for this guy. And then they go back to the police precinct and say, hey, everyone, look for this guy. And then the immune system starts going around and hunting for that guy. And that guy is, in fact, COVID. So that's how it works. So the concern was, well, we're taking these lipid nanoparticles and we're filling your muscle with lipid nanoparticles. Well, is there risk there? Well, one, RNA is short-lived. So the beauty of this is that when you put the RNA in, it will express some protein and then it will just dissolve because it's RNA. It has that extra oxygen, which makes it unstable. And then the transcript falls apart, but the protein's still there. The protein gets presented. Then once that wanted poster gets sent to the local police department, it doesn't matter if the protein's even expressed anymore. It's there. And now there's one police officer who specializes in that particular criminal. And so then that immune cell will copy itself and start hunting the body. And so that's really where the immunity comes from. So then the concern is, well, wouldn't the RNA turn back into DNA? Well, as far as we understand, that doesn't really happen in mammalian cells. We've observed it happening in some more extreme situations. Like there's like things like African sleeping sickness, which has a very exotic way of regulating its genes where it can take RNA and then it's called reverse translating. So it makes DNA off the RNA and then it incorporates it back into the genome. We haven't really seen that happen in mammalian cells with RNA, at least in my entire career. And the RNA doesn't last very long. Furthermore, we already have drugs that do involve permanently adding DNA to cells. It's called gene therapy. So we're already trying to edit cells and edit the DNA in people who have broken genes that cause very severe diseases. Things like Duchenne's muscular dystrophy is one example.
Is that delivered in a similar way in liposomes?
There are two strategies. Yes, you could deliver it with a lipid nanoparticle. The other way is with a literal virus. So we use what's called an adeno-associated virus, which is a virus that infects other viruses, which is what's interesting about it. So normally, like if you just take a regular virus, you don't want it to just kind of go rampant and accidentally edit everyone's genomes. So what we do is we use it's called an adeno-associated virus. So it's a virus that's dependent on an existing adenovirus infection. So that way we have control over whether the virus actually infects a cell by including critical proteins in it. So that way it can't just run rampant on its own.
Right. So it's like it needs something that the adenovirus has in order to complete
its life cycle. Yes. Yeah. And so because of that, we can control its reproduction. And so then what we do is we will edit a variety of, like, we'll mutate 1 million AAVs, and then we start screening them for their sensitivity and specificity across different tissue types. So we can say, and then what we end up finding is we find, hey, there's this AAV that's really good at infecting muscle, but it's bad at infecting everything else. So then we say, if we have a gene you want to deliver only to muscle cells, we'll use that formulation of an AAV. We can package in the gene, and then we can try to replace the broken gene in that disease.
Well, it's all brilliant stuff, isn't it? It's taking the body's own systems to cure things and replace missing bits of message. So, do you know, I as you were making that very excellent explanation, I could almost see it as an animation. You've got the script there for a cartoon version of what's going on. You know, it's what we've got here is, instead of injecting people with a vaccine, we've got a system for injecting people with the means for their body to make the vaccine. We've stepped back a bit and using the body's own systems to to save the pharmaceuticals from making the vaccines.
Precisely. That's precisely how it works. It's funny you mentioned cartoon, actually. There's a Japanese anime, which I'm a huge fan of, called Cells at Work, which basically describes all of the different cells of the immune system in highly personified humanoid characters running around in an allegorical body that does a really, and it's actually fairly medically accurate. Like medical doctors will watch it and they're like, yeah, this pretty much captures exactly like our textbook knowledge of how these cell types behave.
Yeah, excellent.
I always highly recommend that to anybody who wants to understand, like, what are all the different white blood cell, like, what are they called and what do they do? And it's a really great way of conveying.
I will look that up and put it in the details underneath. Do you have the link to it, or?
Let me see if we can find. I think for a while it was on Netflix, but I don't. It might also be on Crunchyroll, but it's basically an anime. It's just called Cells at Work.
Cells at Work. Great. Thank you. Yeah. Wow. So, does this? Tercia brought in the subject of Dr. John Campbell, who is a sort of anti-MMRA campaigner, and I'm very skeptical towards him because I see him in the same vein as Andrew Wakefield, you know, somebody who's pointing to the necessarily dubious aspects of modern treatments, but in so doing, he's risking, he's scaremongering. He's risking people who don't know how things are tested and how they become suitable for usage by, you know, having been tested first of all in animals, then in human volunteers, and then once they've established efficacy and safety, they're tried in small populations of the general public and rolled out, and all of those stages are attempts to learn more about the medication. But what he's doing, it seems to me, is telling people that these things aren't safe, when of course there is no such thing as absolute safe. But what we need to do is not scare people off who could benefit, who could survive as a result of trying these pills or whatever treatment it is.
Yeah, John, if I can just, I really do hope, so Seth, you might be interested in watching the latest YouTube video that Dr. John Campbell posted. I'm a little bit more charitable towards what he says, maybe because I'm more ignorant. That's entirely possible. But that brings me to something that I think is important, in that the whole. Okay, so first let me say that I had my COVID vaccinations. I had my son was 14 at the time. I was severely criticized because as soon as he was eligible to get vaccinated, I vaccinated. All my kids were vaccinated. So I'm fully vaccinated. In South Africa, we don't have the vaccines available anymore. I don't know why. I don't know what the reason for that is. So I got my second booster, and that was it. But I don't think that John Campbell is similar to the Wakefield anti-vaccine doctor. Firstly, he's had 5 booster vaccines himself, and he's now quite nervous about the possible effects or possible after effects. But that is what is so fascinating to me about this whole science, and how we go about doing science. And that's why it's such an honor to have somebody who really works in the wheelhouse to get it from the horse's mouth. And maybe that is that's the conversation that I'm perhaps even more interested in than the nitty-gritty of the science. But how do we communicate these things? Because it is a fact that there has been accidents where there were medications that initially seemed to do the job perfectly well, and then after some years it did You know, you think of the thalidomide, and the fact that the fact is humans practice science, and yes, there are measures in place to try and prevent harm as far as possible. And I'm definitely not one of those people who just say big pharma is evil and they're out to just make money, although they are out to make money, because why else would they be doing what they're doing? They're definitely not charitable organizations. But to me, that's the part that I find fascinating, because I can see that people could be scared, because the less you know, the more strange these things seem, and the more important quality science communication gets. And when it comes to talking about things like DNA and mRNA, it's really, really complicated. I mean, it's not easy to understand. And I think that's perhaps something that needs to be somehow addressed. I'm not sure how, because science is complicated, and the stuff that you work with, to me, it's like magic. It really does sound like it, because there's so much I don't understand. Do you think that do you feel, Seth, working in the field, do you ever feel that the more you learn, the more you know what you don't know? As in,
well,
not you in person, perhaps, but as scientists.
Thank you for that lovely metaphor of a horse's mouth in the wheelhouse.
Yeah, so it's interesting. I had my 20-year high school reunion last year, and I was speaking with one of my former classmates about the vaccine, and she was saying like, yeah, I'm worried about mRNA and all this stuff, and I'm like, look, I literally like worked for one of the companies that was making the vaccines. I saw what was in the vaccine. Like I know exactly what's going on here. And I can understand that there's some basic heuristics people get into with, you know, apprehension and neophobia, and the fact that there's a little bit of seeing behind the curtain too much. I would use the analogy of like you haven't necessarily witnessed every recipe or every piece of food you've consumed. And for example, sometimes when you're watching like a video of somebody preparing a dish, there can be stages where it looks kind of gross, and you're like, I don't know how this is going to come together. But then eventually, like, it does come together, and it's quite beautiful and delicious. And so now imagine somebody who has never been behind the counter. They've never like gone in the back and watched how the chefs prepare their food, and they're like looking over their shoulder halfway through the recipe, and they're like, well, that looks kind of gross. Are you sure I should be putting this in my mouth? And the nature of the pandemic has, for better and for worse, exposed the entire globe to what the process looks like. So what ends up happening is the entire planet witnesses in real time companies like Pfizer, J&J, or AstraZeneca, and you know, Anthony Fauci sharing all the gory details of like how exactly this stuff works. And it makes them nervous because this isn't what they're used to seeing. They're just used to seeing a pill on a shelf that, you know, that they basically take for granted and trust. They don't have a sense of like how science works. Science is a process of trial and error. And so what'll happen is the moment we test our first hypothesis and it gets falsified, everyone panics because they're like, oh, scientists don't know anything after all, we're doomed. Because they don't understand that that's like, to them, science is just people with authority telling them things that are true, and that's what it is. But they don't know how they get there. They don't understand that the way to get there is basically rigorous, formalized guess and check. That's what science is. It's just we've taken the checking part and made it as formalized and optimized as you can, but it's still guess and check. And so when the first guess fails, everyone panics and starts saying, well, see, you can't trust the government. You can't trust the scientists. They're wrong. I'm going to go take ivermectin. But what they don't realize is that they've replaced guess and check with just guess and don't check.
Yes.
Like, and that's the optics problem. And from a scientific community standpoint, we were caught off guard. We were not expecting to suddenly have to be in the spotlight 24/7 and to do these things that we are normally left alone to do and we're trusted to do. Now we suddenly have to justify every single step in the process and deal with all of these uncharitable interpretations and panic people. And we're not trained to do that. We're a bunch of nerds with cool tech and a lot of money thrown at us. And we're not trained at how to communicate this with the public. And then suddenly we're just thrust in the spotlight and told, like, you know, hey, you need to guess correctly on the first try because we're all watching you and waiting on you, and everyone's stuck inside, you know, for want of you solving this problem. Like
you can't guarantee to do that. And if anybody in the general public had understood Popper, they would know that when something's falsified, when a proposition fails, you've learned more than you would if it was substantiated.
What's ironic is also the vaccines were wildly successful because normally it takes like 10 years and multiple rounds of clinical studies. And these companies have routine vaccine divisions that are constantly struggling to get vaccines to work. And the fact that they were able to like do everything at risk, which means they basically threw all their money on a Hail Mary investment on the off chance that it would work. And they even threw money at scaling up the manufacturing for something that might fail its test. Is a minor miracle that they were able to do that, and that everyone's yelling at them with all these crazy conspiracy theories. And the tragedy of it is that, like, okay, so Pfizer decides to do a Hail Mary pass, and they say, okay, we're just going to go for it, and they say, all right, while we're waiting for the results on our vaccine, our Comirnaty BioNTech candidate, which is the official name of the Pfizer vaccine for those who are unaware. I thought it's a funny name. While they're waiting to see if it passes their tests. So they like, okay, we have to put it in and see, does it show the immune response? Does it show immunity differentials in the populations compared to the controls, and do the rigorous tests? They then took a risk and took a bunch of money and started investing in manufacturing of that very same thing at risk. So if the test failed, they would have been just out the money. So they do all that. They end up making like $100 billion. And what happens? The investors are like, well, you didn't make $105 billion, so we're mad at you. And then like they start, I mean, some other nonsense happens to the company. But the point is that like, I think everybody kind of had the whole process ripped wide open without anybody to walk them through how it works. So like when people come along and say, oh, I'm concerned about DNA and all that stuff, I'm like, if he has his own lab and he has peer-reviewed papers, I'm happy to take a look and take it seriously. But if it's just some person saying, I taught nursing and I'm worried about it, like you're borrowing from some adjacent expertise to gain enough clout to then sell your narratives. I
want to perhaps ask, see if John Campbell actually did post the link to the academic paper, 2 papers. I read them. I read the abstracts. But my problem is half of it I didn't understand. I would love, I should have actually had those links with me now. I'll have to look them up. I actually did read the papers, and there were 2 papers that came out, one earlier this year and the other one last year. And they were published the one was published in PubMed. The other I can't remember. And my dilemma is I am absolutely no anti-vaxxer. I will say that 100% equivocally. I'm still of the opinion that the risk of getting COVID is probably greater than the risk of the vaccine. At least in the short term, it certainly was. I don't think there's any doubt about that. And if I were placed in that position ever again, I would probably still take the vaccine, because firstly, I can't imagine that any pharmaceutical company would deliberately push something into the market.
No, it wouldn't be in their interests, would it?
Knowing it will cause harm, because that would absolutely not be in their interest. So I don't see that happening. But I can see that there could be political pressure to solve a problem, and perhaps being a little over hasty. I can see that happening. And I can also see that, you might want, it works, it seems to be working. And firstly, there wasn't time. I mean, we were in the middle of a pandemic.
Hang on. You're leaping to too many. You're doing your chain of thought at the moment.
No, it's called the weave. Donald Trump calls it the weave. I'm being Trumpian here.
Okay. Well,
yes, but you have predicates in your sentences, so you're not doing it properly.
Okay.
Not as properly as him. Yeah. So let's focus on one thought at the moment, shall we?
Yeah. So Seth, if I sent you the links to those papers that John Campbell posted in his link to his the talk was earlier this week. It was Monday, I think. Whenever you have time, please read them and tell us what your opinion is, because I'm not equipped to really understand those papers, but they are papers.
And I think the point is, as Seth indicated earlier, that he may not be either, because really, unless you are pretty conversant with the level of academia required to understand what is actually being done, and he's an ex-nurse, he may not be interpreting it any better than you are.
Absolutely. And that's why we need to have conversations about these topics. And I think that it's brilliant that we have media like YouTube, like the internet, where a normal person like me can actually ask somebody who works in the field, who really knows what mRNA vaccines do. And one thing I do know is that there have been work done on mRNA vaccines for at least 10 years. I mean, the idea of mRNA vaccines are not novel. Am I right if I say that, Seth?
So before you go into that, Seth, I'd like to reinforce what you said earlier, which was that the pharmaceutical company took a big gamble when they went down the mRNA virus vaccine route, because there was already a known process for making vaccines, and it was arguably unnecessary to come up with a new one which had no track record of success at that time.
So what you said is correct. The LNP modality, like putting like mRNA inside of a lipid nanoparticle, has been around for decades. What ironically and somewhat fortunately happened is that due to the rushed nature of the pandemic, investors and regulators and all the stakeholders were like, I think it's worth taking a risk on trying an LNP vaccine because it's so much quicker to manufacture, and you don't have to sit there and just run through all these different like viral variants or trying to process it that way. And due to the urgency of it, they were willing to actually get the investment in it. So prior to the pandemic, we had LNPs, but all the investors were too neophobic and kind of afraid to like nobody wanted to be the first person to take a gamble on it and actually try using LNPs. Now that the pandemic happened and we had a shockingly successful like event with it, all things considered, LNPs are now like one of the most popular investment strategies for the biotech field. And so it's like, it's been sitting there for 20 years. It's just nobody felt like it. It's like, I don't feel like they weren't willing to like dip a toe in the water, so to speak.
So we can thank COVID for kicking it into action.
There's a lot of silver linings that came from it. Like as horrifying as it was, like we got work from home and hybrid models. We got LNPs for better and for worse. We got more public awareness of how drug development works. I feel like we will need to clean that up. As far as like the John Campbell stuff's concerned, I'm doing some cursory Google searches on it, and I'm seeing that now this is just like the first thing. It could be just some severely biased like criticism, but it sounds like he's mostly looking at kind of population level studies of like, there was a population of people who received the vaccine and then something happened to them that might statistically point to something you could blame on the vaccine.
He's doing like, epidemiology.
Yeah. So that kind of like population level stuff. And without getting into the weeds of like how well you can control for confounding factors, there's a fundamental difference between that and like pointing to a mechanism of action. And I think the one thing that we're forgetting is that we had the biggest clinical trial in human history during the pandemic. We literally tested billions of people with the vaccine. So I think we're well past worrying about anecdotal situations where some small population received the vaccine and then sometime afterwards something bad happened to them. You know, for some whatever confounding reason. And from a mechanistic standpoint, the issue here is that there just isn't, like, I'm not really clear what there is to point to. And the thing I would point out to people who are worried about, well, I'd rather take my chances with the virus. Well, they're the same thing. The vaccine is just instructions for making a piece of the virus in a limited and controlled way so that only your immune system sees it. The virus is just flooding your body with copies of the very same material you're worried about from the vaccine. So if you're worried the vaccine, by presenting a spike protein, is going to enrage your immune system to attack and cause pericarditis, the virus is going to do that tenfold worse. And in fact, that's what's been panned out, that anybody who was at pericarditis risk with the vaccine had a tenfold higher risk with the virus itself. So it's like these are all sort of taking it out of context and cherry picking, counting the misses and ignoring the hits, so to speak.
Well,
and the problem, I was just going to say, and the problem with epidemiology is it's a correlation, but it's not a causation. As you said, there's no mechanism.
Yeah, it's good for our generating hypotheses, but then to turn around and say that, oh, there's a problem and that it's reverse translating it into like DNA that's sitting here constantly pumping out spike protein and then causing your immune system to attack your heart. I'm like, well, give me a mouse model. Show me that that's happening. Like you know, don't just show me a bunch of kids in Singapore who had some, you know, concomitant health outcome. Like.
Okay, let me try to not be the devil's advocate. Firstly, I agree with you 100%. What you said about the risk of taking the vaccine versus the risk of getting COVID, because we know that what is well documented is that many people still suffer from long COVID, and many people have severe after effects that will probably affect them for as long as they live. So. But now I want to ask, let's say it is possible for that, so that mRNA somehow does get into my body's DNA. Is that at all possible? And what's the worst thing that could potentially happen? Could it change my DNA? I mean, I absolutely have no idea. But can you imagine?
You could turn into a mutant, Tercia.
So worst case scenario, worst case scenario would be, let's say
sci-fi.
Let's say you accidentally hijack some as yet unknown reverse transcription mechanism, which has never been discovered before. And that mechanism somehow gets hijacked by transposases in your genome. And then they start randomly inserting the spike protein sequence throughout your genome, then you might have a slight elevated cancer risk because you're just causing damage to the DNA. And then let's say you start flooding your cell with spike protein, you know, just from a mechanistic level, like it would probably kill the cell. If it doesn't kill the cell, it would probably cause the cell to be attacked by the immune system. So then the immune system would just kill the cell anyway. And also this is just in the muscle cell. Like it's not like as systemically accessible. There were some cases where they're concerned about like certain LNPs accumulating in the liver. You know, so they're like, oh, well, could it be, you know, modulating the immune cells in your liver or your hepatocytes? Like, so I'm just going based off of plausibility from my current understanding of how these things work to come up with plausible mechanistic hypotheses. Even then, like these are like it's hard to see this as some systemic, like, you know, thing that's going to like melt you or something. You know, it's more, you know, absolute worst case scenario would be an elevated cancer risk or autoimmunity risk. But we simply have never observed that. Like, and then a lot of the people who are struggling with long COVID usually will have a failure of their immune system to successfully clear the virus, or the virus hides in a nervous niche that can escape the immune system. That happens more because the immune system was not able to prevent the virus from getting there, not because there's mRNA turning into DNA and chopping up your genome. Like that's never been observed. Like
let's scotch the idea that a change to your DNA can materially alter your body shape, form, or any any other of your major characteristics, because as you said, it might cause you a cancer, but in order to mutate in the sense that sci-fi movies want to use the word, we need the effect to have got not into the somatic cells, but into the sex cells, because evolution requires reproduction.
Yeah, from the comic book interpretation, if we want to address that, is, if you're using like X-Men as your standard, where DNA is
Yes.
like ongoing instructions to sustain your body plan. Like,
Yes.
no, it doesn't work where like there's a gene that says continue having a nose, and if you turn that off, your nose disappears. Like,
Okay, so are you actually telling me that I can't get injected with something that will alter my DNA so that I'll become tall, blonde, blue-eyed, and skinny? That's not going to happen?
Not via any mechanism I'm familiar with, despite that separate. There might be some like, I feel like you'd have to do like something like stem cell therapy, where you'd have to like inject your bones with some stem cells that might remodel your bones somehow, which would require mechanisms I wouldn't understand. But yeah, I want to make sure that we're not using X-Men as our standard for how we expect changes in the genome to happen. But at the same time, from a communication standpoint, we, as much as we laugh at that, like people will fill placeholders with what they have. And if all they've ever, when they hear DNA and all they think of is like Bruce Banner or the X-Men or something, and they think changing DNA, they're like, oh God, I'm going to turn into some green mutant horror show if I take this vaccine.
My shirt.
Yeah. We have to meet people where they're at. And if the only education they have is comic books, then we need to start there, ironically.
If I was your president, I would demand that all cinemas, all movie theaters have a sign above the door that says, it's not real,
Well, what's interesting about that is that even as much as people can cognitively acknowledge that it's fictional, what people don't necessarily realize that they do is that our minds do not like having a vacuum. So if you like for example, if I say last week I went to my grandmother's house and I walked into her kitchen and I saw that like her dog was there. Can you tell me, with respect to the front door, did I turn left or right to get to her kitchen? Because it's like, what'll happen is, as we think about it, we'll start to compose maps in our mind just to fill in the blanks out of things that we've already known. And a lot of times people will start filling the blanks and they won't even realize it. Like people who don't have experience with warfare or even getting in a fight or what gunfights look like, what do they use? They go to movies. They assume that it's like martial arts or like John Wick, and they don't know what real combat looks like. And then they see the real stuff is way quicker and alien compared to that. And so it's the same thing. We have placeholders that we use in our minds. Like,
and James Bond never gets hit, but all his bullets strike the mark.
Yeah, he hits, everyone else misses, and so of course that's going to get people into these fantasies of good guy with a gun. So it's the same principle.
But this is the kind of conversations that fascinate me, because I'm not a scientist, I'm not a philosopher either. But what you just said now, that we fill our minds fill in gaps, and if you have lived a certain experience and that's all you know, your mind will fill in the gaps. And although people, and when I say people, sort of the average member of the population, they probably know that Bruce Banner turning green and Spider-Man and shooting webs, they know that's not real. But if we don't know enough to know where that threshold is, it takes quite a lot of research. It took me reading and watching some science videos to realize that, all right, if there were chances of the mRNA vaccine contaminating my DNA with some complicated process, then you'd probably look at, like you said, elevated risk of certain cancers and perhaps some a greater proclivity for certain autoimmune disorders. Nothing like turning green or growing a extra set of fingers. But that thing about meeting people where they're at is really important, I think, when it comes to communication about complex topics like science, because most people, like you say, Seth, we know so little. And even if you have high school biology, which I didn't even have, things change. Information changes so fast. I mean, if you had biology as a subject at school, at high school 10 years ago, I'm sure that textbooks now will have things in them that we didn't know 10 years ago. John?
Yeah, but that's not changing things. That's adding stuff.
It's not changing, but
most of what was in biology textbooks in the 1950s was, by the standard of knowledge at that time, correct.
Absolutely.
But since then, we've found out a lot more. We've discovered protein synthesis, DNA, and so we haven't changed what was known before.
No, it's not changing. But the person who quit biology as a subject 20 years ago, that's where they stopped learning.
Well, I would say, like, that's why I think the central dogma is the minimum amount that I would expect or hope a high school program would instill upon the average person. Like, just that everyone remembers mitochondria is the powerhouse of the cell. That phrase is just a meme. And like every other person knows what RNA is. Like, you know, because when I try to explain what I do, I have to, like, everyone knows what DNA is. DNA is pretty as a common, like, it's just some fundamental instruction that makes up your identity is what most people think of it as. But I'd say every other person would know what RNA is. So like, oh, it's something to do with DNA. I don't know what it is, would be their response. The central dogma of DNA to RNA to protein is like 1950s and 60s. So unless your high school education was prior to that, you would have gotten that, and that's about all that's expected. You know, things in like the 70s would be things like alternative splicing, 80s would be things like polymerase chain reaction, 90s you're getting into like microarrays and how to do DNA sequencing, 2000s you're getting into like sequencing by synthesis, and 2010s you're getting into single cell sequencing. So like I wouldn't expect, like, you know, that to ever be taught at the high school level, and most of that stuff I didn't even learn until I was in grad school. So even in my undergraduate education, most of it was just, what do cells do? Like, it's mostly cellular biology and very basic molecular biology.
Seth, what do you like to do for fun?
I'm a big geek and nerd, so I do a lot of role-playing video games. As I mentioned previously, like I had the anime there. I'm also trying to teach myself guitar. So I'm trying to...
So you have a bent for the musical. And then would you describe yourself as a little bit of an armchair philosopher? I know John said that in the bio you like to engage in some philosophy. Do you like debating anti-evolutionists, which is, in my opinion, as weak a term?
Well, that's more anti-science. I mean, I've tried engaging certain anti-evolutionists, but I find like there is a minimum level of agreement with the dialogue that's necessary for a conversation to happen. When I've tried speaking with young Earth creationists, most of the time they'll just ignore everything I say and then like rail on their favorite straw man ad hom. There are certain people there's a small number of people I've encountered who fundamentally disagree with me, who are at least willing to agree with the rules of engagement. So like, you know, at minimum, try to restate your opponent's position in a way they would agree with, so that you can engage with said position and not just straw man or ad hom your way through it. And I find that's exceedingly rare and difficult to find. But if I do find it, it's very precious, and I try to engage those people as much as possible. As far as like philosophy is concerned, like I try to, I'm not like a philosopher per se, and I find it can get a little pedantic with like people fixating on certain definitions. And I'm more trying to figure out like, what model and framework is someone using? When they use a word, what do they mean by it? And then how can I drill down to what the actual point of contention is, and what are their reasons for arriving at it? And 9 times out of 10, their reasons for arriving at their position are completely unrelated to reason or observation. They're more just like, well, if this wasn't the case, the world would be scary, alien, and bad things would happen, is usually their argument. Like my most recent philosophical topic that I've been trying to like, you know, find interlocutors for in the Unbelievable group is nominalism versus realism. Like, you know, are definitions like true facts in the world, or are they just things we make up? Is everything just names all the way down? Like, does the emperor really have no clothes? And there's a lot of people where they're like, well, of course, you know, A equals A, and a thing is a thing. How could it not be not a thing? And I'm like, well, but we have to distinguish between our conversations about something and
Yes,
the
yes.
object.
Why do they do that? I mean, language is entirely fabricated.
Well, because those have very strong implications for things like the trans debate and the abortion debate.
Yeah.
So when you talk to people about topics like trans, like what is a woman, and abortion, what is a person, those all hinge on having this metaphysical ontology where there's a correct, hard and fast answer, almost a platonic form of what it means to be human or what it means to be a woman, and that they like to fixate on. Well, when I say woman, I really think about my stereotypical image I have in my head of what an adult human female, cis female really looks like. And that what womanhood is that, and anything that departs from that is just not a woman, and you're just delusional. Likewise, if they say what is a person, they think, well, I think a human with human DNA. Well, what's the bare minimum of that? It would be like an embryo that had its sperm and egg fused and their calcium signal event, and that now this is a discrete human, and now because it's a human, it's entitled to human rights, and that's as far as they take it. And it's like, well, okay, but then how do you deal with things like the ship of Theseus or the paradox of the heap? So the ship of Theseus, for those who don't know, is like if you have the ship of Theseus is this artifact, and that as it was aging, they kept replacing planks, old planks with new planks until there's not a single original plank. Is it still the same ship? Now, if it is, what happens if they took all the old planks and reassembled the ship again from the old planks? Which one would be the original ship? You know, you could say the same thing with bands or the paradox of the heap. You know, if you have like 1 grain, is it a heap? No. If you had 2 grains, is it a heap? No. So if you keep adding a grain, 1 grain is going to make it a heap, but eventually it's a heap. So what's the precise number of grains to make something a heap? And there's no answer to that. Or what's there's in a cloud and not a cloud? You know, a cloud is just a increasing gradient of water until it hits a threshold where we can see it. But it's not like a physical object where you could say this atom is in the cloud and this atom is not in the cloud. But they would say, yes, there is a cloud and there is a demarcation, but it exists in heaven and we can't find it. But it exists. And
I like the way they try to distinguish between a mist and a fog.
Yeah. So I've been trying to engage with that particular debate where it's almost a form of objectivism, where what they want to do is they want to take the names that we give to things and the pictures we have for them in our head and say that is the true form of the object, that is the true ontology, and that everything that you're seeing is just kind of a rough approximation of where it lies. We might not be able to find where it lies, but there is in fact some fact of the matter about where the true cloud is, or where the true human is, or where the true woman is. And that's sort of this right-left distinction where some people are like, look, we live in a fuzzy, ambiguous world that we round up to functional categories, or we live in a world with hard functional categories that's just imperfect and we're just unable to see which category every time, but they're there. Like, and then why do you think they're there? Well, if they weren't there, it'd be chaos and everything would be horrible. I'm like, well, that's not a reason to say that they're there. It's just saying that you don't like the idea of them not being there.
So I blame Plato. I don't know whether he actually thought that thought is supreme over observations of the natural realm, or whether that's how generations following him have come to regard what his attitude was. But he set humanity back a big step. Whichever is responsible for that mentality, it's hampering the development of science in particular.
I mean, I obviously come from a bias towards empiricism and observation, you know, which only relies on the assumption that there's a mind-independent reality, but it doesn't have to commit to whatever the nature of that reality is. I mean, I don't know enough about Plato to say like whether I think he thinks forms are mental. I suspect that he thinks forms are just sort of out there in like this distant way. Aristotle thought forms existed within the object in and of itself. And then so there was other forms of philosophy that said like there isn't some like, you know, perfectly shaped imprint of you that's up in heaven that like you are failing to adhere to. That imprint lives within your corpus and person, which is fine, but you're still positing this imaginary thing. You're taking the object in our head and you're superimposing it on the physical correlate that we're interacting with.
Which is backwards. It's just backwards.
It does seem backwards, but I'm learning that there are people who have very strong emotional commitment to that idea because they seem utterly horrified by the idea that there might not be anything there, and these are just words we're making up and images we're putting in our head, and that it's all imaginary.
I'm so sorry that because we've got to a really good part of the chat, but we've run out of time. So maybe we'll pick it up another day if you're willing.
And I
Seth.
didn't even get to talk about free will.
Oh boy.
Good. We don't want to go there. That's a can of worms we don't want to open at the moment.
Maybe Seth would have liked to have gone there.
Another time.
I would need at least an hour.
Yes, indeed.
At least.
Yeah. So I can't tell you who we've got as a guest next week, but I'm sure it'll be somebody interesting. And maybe you can recruit one of your, because I know you've got 2 or 3 fish on your hook. You're trying to reel in.
I do. I do actually have some fish to reel in. To
reel in. Good. Yeah. Let's hope that happens. Anyway, we'll announce in the middle of the week who our guest is for next Saturday. Seth, thank you very much. It's been edifying. That's the only word I can think of that's appropriate for this chat. Thank you.
It's been a pleasure.
Tessia, thank you for your contributions as usual. Stalwart.
Well, I'm just still struggling to despite Seth's brilliant explanation, you know, my test for whether I understand something is whether I'd be able to explain it to somebody else.
Well, that is the way. Yes.
And although I can repeat the words, and I actually made notes, Seth, so you know, if you were to ask me right now, explain to me the difference between DNA, RNA, and I wouldn't be able to say the words, but I have to confess that in terms of the triangle of comprehension, I think I'm really at the very base level. So I have some work to do. Let me see if I can get that anime. Maybe anime is the way for me to figure out how cells work.
We'll put that link in the details below. Yeah. And thank you, audience, too. We didn't get many live ones, but I'm sure we'll get plenty in the podcast. Say bye-bye.
Bye-bye.